Hello everyone, this is Ketishia. In this video, I'm going to go through the AQA 2022 Combined Science Biology Paper 1 higher tier. This is question number 4, which is on bioenergetics. Photosynthesis is an important chemical reaction in plants. Why is light needed for photosynthesis? The light is absorbed by chlorophyll for photosynthesis, which means that photosynthesis requires energy, making it an endothermic reaction. So you could say to transfer energy or photosynthesis is an endothermic reaction. 4.2, what is the equation for photosynthesis? Now the equation for photosynthesis should be carbon dioxide plus water should give you glucose and oxygen. So that's the equation for glucose, which means that it can't be the first one. Second one is incorrect. Third, 6CO2, 6H2O. Glucose plus 6 oxygen, that is actually correct. So that it will be the third option. 4.3. A student investigated the effect of different colors of light on the rate of photosynthesis at room temperature. The student used pondweed in water. A piece of pondweed was placed in red light, then in blue light, and then in green light. Each color of light was the same intensity. Describe how the student could make accurate measurements to obtain valid results for the rate of photosynthesis. Now, before I discuss the answer, it will be good to show you an example of what, if I was to do the experiment, what it will actually look like. Now, this image shows the setup I would actually use. So here I would have the lamp, and I should be able to change the different light bulbs for the different colors. Now I am having a ruler so that I can make sure that the distance between the lamp and the beaker is constant, right? So that it's actually a control variable. I'm using a water bath so that the water inside the tube, in the boiling tube, doesn't get hot because of the light. So again, I'm trying to control the temperature using the thermometer. And the pondweed, which is trapped inside this boiling tube, when it produces oxygen, it should be collected by this gas syringe. So what would you do to make sure that the measurements obtained are valid results? So first you would make sure that the pondweed adjusts to the different colors of light. So you'd allow for about 5 minutes for it to adjust before you take measurements. You would use a gas syringe rather than a measuring cylinder. You could say you could use a measuring cylinder as well. And you would also need to time how long it takes for a specific volume of gas to be given out in order to measure the rate of photosynthesis. So remember, to measure the rate of photosynthesis, you would need the volume of oxygen and then divide it by the time. So volume of O2 produced, divide that by the time, that would give you the rate of photosynthesis. So here's the answer. Allow the pondweed to equilibrate in the light before taking measurements. Second, use a gas syringe or measuring cylinder to collect and measure the oxygen gas produced. And measure time for the oxygen gas to be collected using a timer or stopwatch, and then repeat the measurements and then to calculate a mean. That should give you 4 marks. Next part. A scientist investigated the effect of different wavelengths of light on the rate of photosynthesis. The wavelength of light determines the color of the light. Figure 8 shows the students' results. Figure 9 shows the students' results as well. So you've got figure 8, which is a bar graph. You've got rate of photosynthesis in arbitrary units, and you've got the 3 different colors, red, blue, and green. So remember, this is our independent variable, the color of light. So that's my independent variable, and that's the dependent variable that I'm measuring. The second graph, which is figure 9, shows wavelength in numbers, not as categories. Again, this is the independent variable and that's the dependent variable. Question 4.4. Why are the results for the two investigations presented differently? So why is it presented in a bar graph in the first part, and why is it presented in a line graph in figure 9? Well, here you've got both the independent variable and the dependent variable as numbers, which are continuous variables. Here you've got number and in categories, so you call them discrete variables. So if you have one of the variables in discrete form, different categories, then you'll have to present it in the form of a bar graph. So independent variable in the student's investigation is categoric or discrete, and the independent variable in the scientist's investigation is continuous, meaning numbers. Suggest the range for wavelength of green light. Use figure 8 and 9. Now, if you can see using the bar graph, the one for green light is the lowest, and that's 2. That means that's 1 over there, which makes this 6. So the arbitrary units for green light is 6. So if I look at the arbitrary units over here, 6 should be 6, yeah, over there. So that line is 6. Although it's not exactly the same, I could say I would measure the lowest point from there. So let's say there. Between these two wavelengths, it could be green, or it could actually be starting off from here. So I would say from 500 to 550, 575. Yeah. So from, was it 500? Yeah, 500 to 575. Now, if you look at the marking scheme, it actually gives you a range of this. Allow answer in ranges. So it could be, the starting range could be between those two numbers, 475 to 525. And the end range could be between those two numbers, which I have just picked the right at the edge. So that should give you the 1 mark for that. The lowest is 475 and the highest is 650. So they have taken 475 from here, 475 from there to 650, which is here, to be as green color. Right, that brings us to the end of the question. I will see you in the next video. Thank you.